Okay, hello to everybody. Welcome to week 2, and we are going to discuss now chapter number 3. So let me go into slideshow mode. Okay, chapter number 3 is about bacteria and archaea. So we have already discussed that there are three domains which have basically originated from a common ancestor: bacteria, archaea, and then eukaryota. So in chapter number 4 we will be talking about the characteristics of the eukaryotic organisms or the domain eukaryota. So bacteria and archaea, they fall into the broad category which we call prokaryotic organisms. So let's go and discuss about these organisms. So first question is how bacteria and archaea, they are different from eukaryotes. So bacteria and archaea are different from eukaryotes because number one, they do not have nucleus. They do have genetic material, and it is the genetic material is basically the DNA is present, is just there in the cytoplasm, and that region is called a nucleoid region. Okay, so genetic material is there, but it is not present in a special compartment. We call it a nucleus. So that nucleus is present in a eukaryotic organism, so that is why they are called eukaryotic. Eu means true, karyota word comes from nucleus. So the eukaryotic organisms, they have a true nucleus. Bacteria and archaea, they do have genetic material, but they do not have that membrane enclosed nucleus. So the region where the genetic material is there, that's called a nucleoid region. So this is the main difference. And also there is some difference in the cell walls of the prokaryotic organisms and the eukaryotic organisms. In the bacteria, we will study their cell wall is made of a material called peptidoglycan and some other unique chemicals. And also prokaryotic organisms, bacteria and archaea, they lack some of the membrane enclosed organelles, and eukaryotic cells, they have membrane enclosed organelles in them. Okay. Now we are going to talk in details about the structure of the bacterial cell. So all the bacterial cells, they possess membrane, cytoplasmic membrane. They have a cytoplasm, they have ribosomes, they have cytoskeleton, and they have one or few chromosomes. So this is possessed by all the bacterial cells. Most bacterial cells possess cell wall and a structure coating called glycocalyx. So what is glycocalyx? It's a kind of a surface coating which is made of some polysaccharides. Some but not all bacterial cells possess. Some of them can possess, but not all. Flagellum, pili, and fimbriae, we are going to discuss in detail very shortly what they are. An outer membrane, so this is also present in some of the bacteria but not in all. So outer membrane is present in gram-negative bacteria. This is a very important characteristic of gram-negative bacteria, and this distinguishes between gram-positive and gram-negative bacteria, that gram-negative bacteria have this outer membrane. Gram-positive bacteria do not have this membrane. Nanowires and nanotubes, these are all required for the transfer of some amino acids and other things. Plasmids, inclusions, endospores, and microcompartments. So these are some of the structures which can be present in some bacteria but not in all. So we are going to discuss. So let me zoom in the structure of the bacterial cell so that we can understand in a better way. So this is the basic structure of the bacterial cell. So you can see on the left side are some of the things which are present in all the bacteria. Cytoplasmic membrane. So cytoplasmic membrane is this membrane. So it's present in all the bacteria, and this is made of lipids and some proteins, and it allows things to move in and out of the bacterial cell. Bacterial chromosome, or that region where the bacterial chromosome is, that is called, so this is they're showing this is a genetic material in the bacteria, and this region is called the nucleoid region. Ribosomes, you can see all these things. These are basically some kind of complex structures, and we normally call them protein factories because on the ribosomes proteins are made. So you can see in the bacteria ribosomes are there. Then cytoplasm, this is all the cytoplasm, this region. So cytoplasm is basically the jelly kind of a material which is present It's actually a characteristic of all the cells. So all the organelles, or all whatever you see in the bacterial cell, bacterial cells do not have the organelles. Everything is embedded in on a cytoplasm, in the cytoplasm. So all these you can see, your genetic material, ribosomes, everything is embedded in the cytoplasm. So these are the things which are there in all the cells. Now let us see some of the things which are present in some bacteria. Okay, S layer. Now what is S layer? This is the S layer. It's basically a layer of some proteins. It's a monolayer of proteins. It's used for protection and the attachment. Now you can see here these short hair-like bristles, these extensions you can see. This is called fimbriae, and their function is for the adhesion. So the bacterial cells, when they have to first of all invade some cells, like if suppose you're getting infected with a bacteria, and suppose they're infecting your respiratory system, first of all these bacteria, they need to attach to our respiratory epithelial cells. So how do they attach? They attach themselves through these structures called fimbriae. So they help in the attachment, or scientifically we call that word is adhesion. Now outer membrane, as I said, this is a membrane. This is a extra membrane which is very similar to the cytoplasmic membrane, which is there in all, which is there in all the bacterial cells, the cell membrane. This is extra membrane which is present in some of the bacteria, and gram-negative bacteria, this membrane is there, and it can have some specialized polysaccharides like LPS. Cell wall is there in some bacteria but not in all bacteria, and it is made of a special material called peptidoglycan. Now the cytoskeleton means the skeleton of the cell. So cytoskeleton is just like our body has a skeleton, and the skeleton is made of bones and ligaments and all those things, muscles. Similarly, every cell, right now we are talking about the bacterial cells, they have a cytoskeleton, and that is made of some protein fibers. Pilus. So pilus or pili we call it. It's a kind of a appendage, and this is basically helps in the process of conjugation, in which two bacterial cells, they can come close to each other, they can conjugate, and they can exchange genetic material with each other. So that is the pilus. So this is the pilus. Then glycocalyx. Glycocalyx is basically a kind of a coating or the layer which is external to the cell wall. Okay, it serves protective, adhesive, and receptor functions. Okay, next is inclusion and granules. So basically you can see these are inclusions or inclusion bodies or granules, and they are used by bacteria for storing nutrients like fat, phosphate, or glycogen in them. Next is bacterial microcompartments like this. They are basically for storing some of the enzymes in them. Okay, plasmids. Plasmids are basically circular. They are extra chromosomal. So you can see this is the main genetic material of the bacteria. They do have some extra chromosomal genetic material, and they are called plasmids. And these plasmids, they also carry some of the very important genes, especially the antibiotic resistance genes and all that. They are carried on the plasmids. Flagellum. So flagellum is, this is the flagellum. This is a long appendage, and it is used by the bacteria for the movement, for the motility. Okay, some of the bacteria can also have these some of these features like nanotubes and nanowires. So they are basically membrane extensions that allow bacteria to transmit electrons or nutrients to the bacteria. So this is again used for the these are special structures which are used by bacteria for the transfer of, or for the exchange of some of the nutrients. Endospore. Endospore is basically a kind of a dormant body. Whenever the conditions are not very good, not very favorable, at that time the bacteria can form endospores, and they can resist these harsh conditions. That is what the endospore is. Okay, now we're going to talk about bacterial shapes. They're very important, and we will be talking about all these things more in the lab. Also, you will be actually making some. You will be doing some of the staining. You'll see various bacterial shapes. So usually bacteria, they're unicellular, but they can live together as colonies. The bacteria, they're very small. They're microscopic, as you know, and their size in the range of 1 micrometer. So let's talk about the different shapes of the bacteria. Let me zoom it again. Okay. Now you can see here the first very important shape is coccus. Okay, we can call it a cocci or coccus. Usually they are spherical in shape. Okay, so you can see this kind of a shape. This is all spherical, circular shape, or you can also call a ball shaped. Okay, so this shape is called coccus or cocci. There can be little different modifications of the shape as it's written here. Cocci can be perfect spheres, but they can also exist as oval or bean shaped. Okay, but mostly they are spherical. They're called coccus, and the typical example is staphylococcus. Then they can be rods. Okay, like you can see they are like cylinders, and rods are also called bacillus. And something very important to know is there is one genus of the bacteria which is also called bacillus. So especially when you have bacteria which has a shape of a rod, we also call them rods, or we also call them bacillus or bacilli. And if the rod is short and plump, like if the rod is very small and it's little thick, we call them cocobacillus. Next shape is vibrio. Now what are vibrio? They are usually singly occurring rods that are generally curved. So if you see these structures like that, they are like a rod, single rods, and if they're little curved, we call them, the shape is called vibrio. Then spirillum. Spirillum is slightly curled, or they have a spiral shaped body. This shape is called spirillum. Then spirochetes, this is another spiral shape. Okay, and they have a periplasmic flagella. So this flagella is quite inside the bacterial cell, and it is in a periplasmic space. And then we have another shape, branching filaments. They are basically the bacteria with rod shapes, but some bacteria can produce branches of the basic rod structure. So they are basically like a rod, but they can form like these kind of branches. So few bacteria produce multiple branches of a basic rod structure. This is a basic rod structure, and you can see how they're forming branches. Okay, so these are different shapes of the bacteria. Now let's talk about the arrangement. First of all, we are going to talk about the arrangement of the cocci. Cocci are the spherical bacteria. So they can occur as single, they can occur in pairs, they can be in the group of 4. So when they are in pairs, we call diplococci. When they are in a group of 4, we call them tetrads. They can also form irregular clusters. Example is staphylococci and micrococci, or they can be in chains. Okay, then the typical example is streptococci, or they can be in the packet of 8 or 16. Then they're called sarkinas. So you can see here. Just a second. Oh, I'm sorry. Okay, yeah. So you can see here, like these two cocci, they are diplococci. You can see the cocci in chains, streptococci, in tetrads, like they can exist like in this tetrads, or they can be in the packet of 8, 16 like this. They're called sarkinas, or they can be irregular clusters like this. The example is staphylococci and micrococci. Now we're going to talk about the arrangement of rods or bacilli. They can exist as single. They can exist as diplococci, which means a pair of cells, 2 rods together, diplobacilli. Streptobacilli, when the rods, they exist in chains. Palisades, when the chains, these bacilli, they are in chains and they are partially attached by a small hinge region at the ends. So they are basically chains, but they are attached. Spirilla, so they are occasionally found in short chains. Spirochetes, they rarely remain attached after cell division. Okay, now we're going to talk about, we have just seen some of the structures. Now we're going to talk about a little bit in more detail. So the bacteria, as we said, they can have flagellum. The flagellum is basically for the motility, for the movement of the bacteria. The other structures are fimbriae, pili, and nanotubes and nanowires. They are usually for attachment to the surface or to the cells wherever they are trying to attach. So they are for adhesion and also for the exchange of the material, of the exchange of the genetic material or exchange of nutrients. Now, on the basis of flagellum, so this flagellum can be arranged in different ways. Okay, so it can be a polar arrangement. Polar means when the flagellum is attached at one or both ends of the cell. Now, the flagellum can be monotrichous. If the bacterial cells have only one flagellum, then we call it a monotrichous. They can have amphitrichous, which means flagellum is at both poles of the cells. That's amphitrichous. Peritrichous, when the flagellum is randomly dispersed throughout the surface of the cell. That's peritrichous. What is lophotrichous? Small bunches or tufts of flagella emerging from the same side. Let me show you now. So this is monotrichous flagellum. This is peritrichous. So this is monotrichous, one flagellum. This flagellum is all over the surface. Okay, you can see all peritrichous. This is amphitrichous on both sides. This is lophotrichous. Okay, now this is fimbriae. You can see fimbriae, these are short appendages. They are short, small bristle-like fibers. You can see these are, and they basically help in the adhesion. Okay, so the bacterial cells, if they have to infect some host, first of all they need to attach to the host cell's surface, and they attach through the fimbriae. Now, as I said, pili and pilus, they are used in conjugation. The two bacterial cells, they conjugate with the help of these structures, and they can exchange genetic material. Nanotubes and nanowires, they are basically the extensions of the cytoplasmic membrane, and they are used as channel to transfer the amino acids, and amino acids is a kind of a nutrients. So this is the conjugating process. You can see these are two bacterial cells. Okay, so this is short appendages around them. They are called fimbriae, and you can see these are the pili. So these are the pili, and they are interacting. These bacterial cells are interacting or conjugating with each other through these pili or pilus. Now, what is S layer? S layer is basically a layer which is made of some proteins, and this usually layer is made by bacteria in some hostile environment. Glycocalyx, it's a coating of repeating polysaccharides. Polysaccharide is a form of a sugar, or glycoproteins. Glycoproteins are basically proteins with some sugar chains on them. So glycocalyx is a kind of a coating over the surface of the bacteria. Now this glycocalyx can be in the form of a capsule, or it can be in the form of slime layers. Okay, these bacterial cells, they form capsules, or they can also form these slime layers. So they are both kinds of glycocalyx. Now what is a slime layer? It is loose, and it protects the bacterial cells against the loss of water and nutrients. And what is a capsule? It's tight, okay, and it is dense and it's thick. And if you will see, we may do in the lab that if we have some bacterial cells, when they form colonies on the agar, if those bacterial cells, they make a capsule, those colonies are little sticky. So now you can see here, this is the cytoplasmic membrane which is there in all the bacterial cells. Outside the cytoplasmic membrane is the cell wall, which is made of peptidoglycan, and then there is a S layer, and then there is a glycocalyx. So this is basically showing you some of the colonies which are growing on the agar. You can see they are producing a kind of a sticky material. You can see that with your eyes when the bacterial cells which are capsulated, when they grow on the agar, they form little sticky colonies. And this is the kind of a negative staining which is done, and you can see this is all the capsule which is produced by these bacterial cells. Okay, so basically what is the function of capsules, why some of the bacteria produces the capsule, and what is the character of a capsule, what advantage does a capsule give to the bacteria. So it is a formed capsules are formed by many pathogenic bacteria. Some of the bacteria which are very pathogenic, which can cause diseases, they produce capsules, and they protect against the process of phagocytosis. So phagocytosis is a process which is done by our own immune system to kill all these infectious things which are entering our body. So it's a kind of an immune response, and the bacteria which produces these capsules, they can protect themselves against this process of phagocytosis. Then the cell envelope. So basically it lies outside the cytoplasm, and it so what is considered under the cell envelope? So cell envelope basically is composed of 2 to 3 basic layers, which includes your cell wall, cytoplasmic membrane, and the outer membrane. Okay, so all these three things make the cell envelope. So you can see that there is a difference positive and gram negative bacteria. So basically this is the gram positive bacteria, this is the gram negative bacteria. So this is the cytoplasmic membrane in the gram positive bacteria, and this is the cell wall in the gram positive bacteria, which is very thick, and it is made of peptidoglycan. And this is the cell envelope of gram negative bacteria. This is the cytoplasmic membrane. This is a very thin layer of the peptidoglycan, which is a material which makes the cell wall. So the cell wall in the gram negative bacteria is not very thick, but gram negative bacteria, they have a extra outer membrane. So this outer membrane is there in gram negative bacteria, and it is not there in the gram positive bacteria. And this is the main difference. That's why when we do the gram staining, these cells, they stain purple, and these cells stain pink in color. So we will be talking more about gram staining in the lab, and the real reason lies in this structure, that the cell wall which is made of peptidoglycan, it is very thick in gram positive bacteria, and it's very thin in gram negative bacteria. So cell wall basically determines the shape of the bacteria. It provides a strong structure support, and it prevents the bacteria from bursting, because cell wall basically prevents the entering of the water inside the bacterial cell and prevents its bursting. So there are all these antibiotics, especially the penicillin antibiotic. This category of antibiotics, basically they weaken the cell wall of the bacteria, and as a result of that, the water can enter into the bacterial cell and the bacteria bursts, and that's the way it dies. Okay. So peptidoglycan is the material of which the bacterial cell wall is made. It is basically a compound which is composed of the sugar chains, which we call them glycan, and they are cross-linked by short peptides. Okay, so you can see all these are all sugars. Okay, these are all sugars, and in between they are cross-linked by the peptide. So that is what the peptidoglycan is, and the peptidoglycan is a material which forms the bacterial cell wall. And the bacterial cell wall also contains these another acids called taconic acid and lipotaconic acid. And they function in cell wall maintenance and enlargement, and they contribute to the acidic changes on the cell surface. As I already said, the gram negative bacteria, they have a thin sheet of peptidoglycan. Okay, the gram-negative bacteria, they have a thin layer of peptidoglycan. The cell wall is thin, but they have extra outer membrane. And this extra outer membrane has some special material in it. It has lipopolysaccharide. Okay, so it's a polysaccharide with some lipids in it, and it serves various purposes. And also the outer membrane of the gram-negative bacteria also has some specialized proteins in them, and these special proteins we call them porin proteins, and they are especially they are basically the membrane channel proteins. So what are membrane channel proteins? They are the proteins which are present in the membrane, and they help in the movement of things in and out of the cell. So they are special membrane channels that only allow certain chemicals to penetrate. Now the cytoplasmic membrane, which is there in all the bacterial cells, it is basically made of lipid bilayer with proteins embedded in them, and it serves for energy reactions, nutrient processing, and synthesis. So basically through a cytoplasmic membrane, it's made of lipids, and it has proteins in them, and because of all these proteins embedded in the lipid bilayer, this membrane is acting a site for various energy reactions, various nutrients are getting processed, and various synthesis is taking place. And basically this membrane is selectively permeable, and it is helping in the movement of things in and out of the bacterial cell. So regulates transport of nutrients and waste. So things are moving in and out of the bacterial cells through the cytoplasmic membrane. Okay, we have already discussed the difference in the cell envelope of gram-positive and the gram-negative bacteria. So the outer membrane of the gram-negative bacteria, which is extra in them, gram-positive don't have it. So basically it acts as a great barrier, and because of that, these bacteria, they are more resistant to the antimicrobial chemicals. Okay, so it's more difficult to kill the gram-negative bacteria as compared to gram-positive bacteria. But there are some of the alcohol-based compounds which can dissolve, because that's all made of, the outer membrane is made of some lipids. So you can use some of the alcohol-based compounds, and they can dissolve the lipids which are there in the outer membrane, and they can cause the damage to the gram-negative bacteria. So that is why you must have seen sometimes these alcohol swabs are all used to clean the skin before any medical procedure. The reason is alcohol can take the it can dissolve the lipids which are there in the outer membrane of the gram-negative bacteria. Now the cytoplasm in the bacterial cells, it basically is made of 70 to 80% water, and this cytoplasm also has some sugars and amino acids and salts in them. Basically the most of the bacteria, they have one single chromosome. They have one single chromosome, and that is present in the cytoplasm, and that region we call it a nucleoid region. And some of the bacteria also have some extra chromosomal DNA in them, which is circular, and we call them plasmids, and they're carrying some of the very, very important genes on them. Now what are ribosomes? The ribosomes, we usually call them as protein factories, and they are composed of rRNA and the proteins. So ribosomes are kind of complex structures, and they are made of two components. They're made of proteins and rRNA. Now what is rRNA? R stands for ribosomal RNA. Okay, RNA is another kind of a genetic material, and it is there can be different kinds of RNAs. There's the mRNA, there's the tRNA, there's the rRNA. So rRNA, R comes from ribosomes, and rRNA is one of the component of the ribosomes. And the ribosomes, they are basically different in prokaryotic and the eukaryotic cells. And these ribosomes, they are made of two subunits. One is small subunit, another is large. In the bacterial cells, the small subunit, its size is 30S. S is a kind of a unit of the size of these subunits of the ribosomes. Large subunit is 50S, and together they make 70S. The size of the ribosome in the bacteria is 70S. Even in archaea, the ribosome is 70S. In eukaryotes, it's 80S. So let me show you. So in the bacterial ribosome, this is a large subunit. Its size is 50S. This is the small subunit. Its size is 30. So this is the small. This is the way they assemble. This is the small subunit. This is a large subunit. You can see together the size is 70S. Now we have already talked about inclusion bodies in the bacteria. Inclusion bodies are basically used for the storage of the food, or they can also pack some gas in these vesicles for the bacteria can then float. Okay, they can also store some iron oxide, and they can give these, some of the bacteria, they have magnetic properties, and it's all because of these iron oxide in these inclusion bodies. Bacterial microcompartments, they are basically having a lot of enzymes in them, and these enzymes basically help in biochemical pathways. Cytoskeleton, as I said, bacterial cells, they also have a skeleton, and this skeleton basically contributes to the cell shape, and it is basically made of some protein filaments. Bacterial endospores. So bacteria, when the conditions are not good, the bacteria form these dormant bodies, okay, just to protect themselves and to survive. And these endospores are produced by some of the bacteria like Bacillus, Clostridia, and Sporosarcina. Okay, so endospores are like dormant bodies, and then they can, when the conditions are good, they can form the vegetative body again. And what is vegetative body? Which is a metabolically active stage. Now these are some of the bacteria which form endospores. Bacillus anthracis, which is a causative agent of anthrax. Clostridium tetani, which is cause of tetanus. Clostridium perfringens, cause of gas gangrene. So these are all important from medical point of view, because these are some of the bacteria which can form endospores. And the bacteria which can form endospores, they are more difficult to treat, because whenever the conditions become bad for them, they are going to form spores. Clostridium botulinum causes botulism, and Clostridium difficile, which causes gastrointestinal disease. Clostridium difficile. Okay, the other prokaryotes is archaea. So this is earlier bacteria and archaea, they were in the same group, but as the science advanced more and we came to know, because morphologically they are very similar to bacteria, and then when we came to know about more techniques and more science developed, we came to know about a kind of a sequencing which we call rRNA sequencing. And rRNA is one of the component of the ribosomes. On the basis of this kind of sequencing, scientists came to know that this archaea, they have, although morphologically they are very similar to bacteria, but they have some of the, on the basis of this kind of sequencing, they came to know they are more related to eukaryotes. Okay, so share RNA sequences not found in bacteria. On the basis of this sequencing, they came to know they resemble more eukaryotes and less bacteria. So that's why, but they are morphologically they are very similar to bacteria. The structure is similar to bacteria. So now they have been now categorized in a different group. So these archaea, they have unique sequences in rRNA. They also have some unique membrane lipids. So they also have their cell wall components are also different. Pylon proteins are also different. They have all these things unique, and that's why now they are in a different group. And they can, we also call them extremophiles, which means they can really tolerate very harsh temperature conditions. They can also tolerate very high salt or acid conditions. They can also live on sulfur or methane. And this is a table which shows you, which compares you the characters of all the different groups, so you can go with them. Okay, so that's all for this chapter.